Thank you very much. Yeah, and thank you all the organizers for the invitation to such a great
We don't
event.
hear you very well. Please, a little bit closer.
Now
Now
it's fine?
better.
Okay, thank you. Yeah, and it's especially pleasure for me to talk after such an insightful talk by Roman Yefremov, given that I started my research career in his lab. So saying that, let's switch to my talk about spatiotemporal identification of binding sites with computer vision. Just basic notions. So what's the binding site? We have the target, which is in most of the cases is the macromolecule, typically the protein macromolecule, and we have a ligand, which can be a drug candidate, or the endogenous molecule. And so when they interact, the interaction takes place not over all the surface of the protein, but in a specific spatial region in the protein structure. And this spatial region called the binding site. So in drug discovery, that's an important concept. And there are two types of the binding sites. The orthosteric binding site is the binding site where the endogenous molecule bind, like the hormones or other small molecules, and the allosteric binding sites, which are topologically distinct from the orthosteric binding site. Both of them are a very interesting object for research. Regarding the orthosteric binding sites, they are typically the classical hotspots for the drug discovery campaign, for the molecular docking and screening. We still do not know all orthosteric binding sites in our proteome, for example, for some orphan proteins. And another important thing that orthosteric binding sites may carry missense mutations that affect the therapeutic effect. On the other hand, we have the allosteric binding sites that will work thanks to this phenomena of the allosteric modulation, when we affect one region in the protein structure, and through the allosteric modulation, we affect the entire behavior of the protein, its signaling properties, for example. So the allosteric binding sites, they are can be used to design more selective, safer, more effective drugs, and typically to discover the negative or positive allosteric modulators. Next thing to notice is that we do not have the entire map of orthosteric and allosteric binding sites still, even for the well-known proteins. So this is just a recent example published in Nature Chemical Biology about the beta-2-adrenergic receptor, a pharmacological target with long history. So here you can see the picture of this receptor. Upper part corresponds to the extracellular part, bottom part, the interior of the cell, the membrane shown here. And so the orthosteric binding site, it's located somewhere here. You may see the gray sticks corresponding to the orthosteric ligand. But here, just on the right side of the protein, you see this compound AS408, which is the newly discovered negative allosteric modulator. And it was discovered just a few years ago. And here in this plot, you can see how it affects the biological response of this protein. On the x-axis, we see the norepinephrine concentration, which is the orthosteric ligand. By color, we see the increasing concentration of this allosteric ligand. And we see that while we increase the concentration of this allosteric modulator, the biological responses decreases. Another interesting example is the EGFR, the kinase domain of this protein. And we know that it works at the asymmetric dimer. And recently, this work was published in Nature, the mutant selective allosteric binding sites were discovered for this protein. So here in green arrows, you may see the orthosteric ligand bound to the orthosteric binding site. And with the orange arrow, you see the allosteric ligand bound here. Also, EGFR is the well-known pharmacological target, but only recently such type of allosteric modulator was discovered. And so we apply the computer vision approaches to detect the binding sites. The first question, what does computer vision have to do with it? Well, if we think about the problem, it actually resembles the object detection problem in the computer vision. So on the one hand, we have a picture, which is 2D image. It has the dimensions, height and width in pixels. It has the RGB channels that encode the image. And the goal is to locate a cap on this example. So to construct the bounding box around the object we are interested in. On the other hand, we have the protein, which is the 3D image, can be considered as 3D image. It has dimensions in the physical scale, the nanometer scale. We can represent it with the atomic channels rather than the RGB channels. And the goal is to locate the bounding box, the 3D bounding box in this case. So the main question is, how do we colorize the protein? What are the channels, these atomic channels in our case? So we came up with the atomic typization where the color corresponds to the specific atomic type in the protein structure. So here you can see on the right our coloring code. For example, the sulfur has a separate channel, the nitrogens have separate channels, oxygens and carbons. And once we introduce this like color scheme, now we can convert the initial protein structure into the 3D image, which is the 4D tensor. So once we've done this, now we can apply the computer vision approaches in the same way as it is done for the pictures. So we process, in our example, we process this 3D image with the 3D convolutional neural network. So the protein structure is in the input, and in the output, we predict the geometric center of the binding site along with the probability score or the confidence score corresponding to this binding site. The next interesting thing that the binding sites generally they differ with respect to the ligand type or the macromolecular type, and we can consider different types of the binding sites as different objects to detect. So here, for example, the binding site for the small molecule in most of the cases corresponds to some cavity, but already for the peptide binding sites, it's different. So they are very often elongated in shape. And if we consider non-protein pharmacological targets like RNA or DNA, these are completely different atomic structures. So we do need different colorization scheme. But otherwise, the method is the same, and we showed that it works and performed better than the state-of-the-art approaches by the time of these publications. So here's just the metric. As the metric, we use the average precision. We called our approach the ByteNet, and you may see that it's performed better compared to the other approaches listed on the x-axis. Another important thing related to the previous talk, that the proteins, they are dynamic by nature, and in the experimental structures, we do have the static structures. So we're talking about crystallographical structures or the cryo-EM structures. So the problem is that in these experimental structures, it's very easy to overlook the binding site because the protein is dynamic and the binding site is the dynamic. In the static structure, it can be closed, it can be collapsed. So we do need to take into account the dynamic information. And again, and we do not have experimental data for this, but we do have the computational tools for this, like Monte Carlo or molecular dynamic simulation. So this is the example of the GFR I showed you before, asymmetric dimer, and in the blue subunit here, we have the two ligands with the magenta one is the allosteric ligand. In the pink subunit, we have only the orthosteric ligand. So the pink subunit correspond to the unbound structure with respect to the allosteric ligand, while the blue one correspond to the bound structure. And so if we model this transition from the unbound to bound structure, we may see that the method can predict the opening of the allosteric binding site from the simulation. So the sphere corresponds to the prediction, the size of the sphere, the radius of the sphere corresponds to the score of the prediction. And well, let me start it again. In the very beginning, we do not see the pink sphere here, or the radius is too small. But once we model this transition, unbound to bound transition, we see that the confidence is increasing while the structure is slightly modified towards the bound state with respect to the allosteric loop. Another example for the transmembrane receptors. So this corresponds to the molecular dynamic simulations of a GPCR, fully atomic. So the membrane, the lipids, the water, the ions are not shown here for simplicity. The method is applied only to the protein structure, so the method does not see any other molecule. And this animation shows that we can detect the binding event using the molecular dynamic simulation. So the method is applied on each time step of the molecular dynamic simulation. You see the lipid molecule here, and once the lipid binds to the receptor, we see that the method detects this binding. But the method does not see the lipid itself. So it highlights the region where the lipid binds. And once the lipid is bound, it stays there, and the method also sends this conformation-specific binding event. The next example is done with Sanofi. It's based on the KRAS case study. So KRAS is another well-known oncological target, and the problem with KRAS is that it's very dynamic. And for the long time, we knew only the orthosteric binding site or nucleotide binding site. The problem is that the nucleotide has very high binding affinity, like picomolar. So it's almost impossible to discover a drug that competes with such high binding affinity. So the problem for a long time was to discover the allosteric binding site and the allosteric ligand. And using, again, the molecular dynamic simulation provided by Sanofi, we showed that we can detect the all types of the binding sites known for this target. The nucleotide apparently, and to others like switch 1,2 and switch 2 binding sites from the molecular dynamic simulation, starting from the unbound conformation, which is very important. And the last example is about the SARS-CoV-2 spike. This is done in collaboration with the Institute of the Bioorganic Chemistry. So we used the long-term molecular dynamic simulation provided by the David Shaw research company. It's like microsecond simulations of full atomic simulations of this huge system. So we detected the cryptic binding site in this case and ran the virtual ligand screening campaign against the found cryptic binding site, shown here. Well, we purchased the most promising hits, and in Institute of Bioorganic Chemistry, they used this pseudovirus assay in order to test this compounds like luciferase assay. And we do found the 3 hits that inhibit the infection process. So these are the preliminary studies. So that I'm going to work. Okay, so finally, I would like to say that our approach, the binding site, is free for the community. You can use the web server. It has like thousands of users already. Feel free to contact me for collaboration if you're interested in. I would like to thank the organizers once again for such a great event, and I want to acknowledge the collaborators from Fusion Pharma, Sanofi, from the Institute of Bioorganic Chemistry, of course the HPC facilities, and the group I lead in Schooltech, which are basically listed here. Okay, so thank you very much, and I'm open for the questions.
Thank you, Dr. Popov, for your nice presentation. Unfortunately, I didn't see any question, so I think we can move to next speaker.
So sorry, Afina.
Didn't see your mail.
Actually, I asked something and
I'm
Just,
sorry, yes.
Just to remind that we
Yes,
have to
since-
follow the schedule. So the
okay,
next speaker should start exactly at her or
Okay.
his time.
But there is one question from you. I don't know if it's to everyone.
No, it's questions to Piotr Popov. Not
Since
everyone.
our knowledge about the biological function is limited, why do not suggest that at least some allosteric binding sites may have endogenic ligands which are still unknown? And what kind of and another
No,
question?
it's another
It's
question.
another
but-
question. Maybe it's he can answer to both together during the time.
One by one, I think. One by
Okay.
one. Okay, so for the first question, I think it's more about the definitions. So yeah, that's true that there is no like strict separation between the orthosteric and the allosteric with respect to the endogenous ligand. But the common definition is once we know the orthosteric ligand, everything that is bind to the topologically distinct binding sites calls the allosteric binding sites. And what is bound to the allosteric binding sites called the allosteric ligand. It's true that it is possible that endogenous molecules may bound into the both places. That's of course true.
Thank you. Next question is from Pavel Polishuk, who asked what kind of protein structures we used for model training apostructures or structures from complexes.
Okay, so for the training, we used the bound structures for sure, because it's, well, apparently you can, let me think. Yeah, the bound structures, because the conformation is important. If we use the apostructure, the binding site there may be collapsed or may be closed. So when we train the model, we want to be sure that the binding site is in the open conformation that is capable of binding. Okay.
Dmitry Solotkin asked, follow up to Dr. Paroikov question. It could be a problem of unknown unknowns.
Well, exactly, yeah. So we can only speculate here.
Vladimir Sulimov, do you think that we can believe results obtained on the long MD trajectory microseconds? How did you check the accuracy in?
Yeah, well, of course, we cannot just blindly believe to MD, and we should be always skeptical about the simulations that we obtain. But that's why we do need the experimental validation in the first place. So we do the modeling. We believe that we did everything right, at least in our capabilities. But then we purchase the compounds, we ship the compounds to the experimentalists, and they validate the result experimentally. Generally speaking, it still doesn't prove that the modeling was right until we get the atomic structures of the ligand bound to the critic binding site that we predicted. But this type of the experiment it's very expensive, and only few labs, I think, in the world can do this.
There is another question from Pavel Polishuk, but I don't know if they have time. How suitable or confidence the developed tool for prediction of sites on apoproteins or cryptic site identification if it was training only on complexes?
Yeah, that's why that's why we try to use the dynamic information exactly for that. If we have in order to catch the binding site starting from the upper structure. That's what I showed for the EGFR example when we modeled the transition from the unbound to bound. Yeah. Okay. Thank you for the questions.